What I'm presenting today is a work that I've been developing for quite a while now, and I have to say it's sort of a continuation of my work into how various types of diseases, especially sort of very elusive diseases, are conceptualized through the use of neuroimaging. So far, much of my research has focused on what used to be called hysteria and what is now called functional neurological disorder. And now I have sort of expanded the scope and I'm looking at Alzheimer's disease. And although there are sort of huge differences between these diseases, of course, one thing that they both have in common is their heterogeneity and elusiveness. And what I want to do here today is to discuss emerging research on machine learning algorithms for predictively diagnosing Alzheimer's disease in non-symptomatic individuals using structural neuroimaging data. And basically, my main argument is that the process through which predictive diagnostic separability between healthy subjects and future AD patients is constructed through comparisons of neuroimaging data during training and evaluating new ML algorithms is itself reconfiguring the current boundaries between healthy aging and dementia. And I also want to suggest that this reconfiguration has important epistemic and ethical implications. So I know that I'm here in the sort of classical philosophy context, but my approach is more sort of aligned with science and technology studies. So I examine technologically mediated operations and semantic negotiations in the development of these algorithms. And I focus primarily on AI in the making, so how algorithms are constructed and trained in practice. And the specificity of my inter or transdisciplinary approach, as you want to call it, is that I extend a.r.t.e.s approach through media theoretical perspectives on meaning construction. Primarily, I draw on media theorist Ludwig Jaeger, whom I will introduce shortly later. And sort of this media perspective allows me to focus more specifically on images as sort of mediating tools. Now, the structure of my talk is the following. First, I will discuss current clinical ambiguities and diagnostic difficulties of Alzheimer's disease, which I argue is a motivation for, but also the source of epistemic contingencies of the emerging ML research into predictive diagnosis. Then I will sort of provide a brief overview of this emerging research into ML models for predictively diagnosing AD based on neuroimaging data. Finally, sort of the main part is my analysis of how in select studies, semantic difference between health and future disease is established through training and testing new algorithms, which sort of jointly is called ground truthing as a process. And finally, I want to discuss some of the epistemic and ethical implications of the thus established semantic differences between health and future disease. Which brings me to the first part of the talk. So regarding the clinical background, I'm well aware that I'm talking within this context of this larger network where a lot of different talks have already been given on Alzheimer, but I still sort of want to emphasize certain points which might have been repeatedly said throughout this series of lectures. But nevertheless, they're sort of important for contextualizing the kind of studies that I want to analyze. So I hope you don't mind if you hear some of these aspects repeatedly. So first of all, you all know that Alzheimer is a neurodegenerative disease, a progressive one, and it accounts for up to 80% of all dementia cases. It is characterized by the loss of brain function and structure, which then in turn lead to progressive cognitive impairment. And 98% of drug trials resulted in failure since 2003, which is also sort of a big motivation for why the research that I'm going to talk about also sort of emerges. There is currently no known cure, and although there are a few treatments, they're only disease modifying. So they are aimed at slowing the progression of the disease, but not at reversing it. And the death is sort of an inevitable consequence, which happens relatively soon after the diagnosis. Now, the greatest risk factor is old age. Typically, it is said people above the age of 65 are at risk. And the major sort of challenge is differentiating between old age as sort of part of normal aging and old age as risk factor for Alzheimer. Now, there's an immense clinical ambiguity regarding Alzheimer because there's a lot of heterogeneity, both the speed and trajectories and the symptoms of cognitive decline differ across patients, but also sort of at the level of neuroimaging, the topography of brain pathologies also vary considerably. This is the reason why some researchers have also argued that Alzheimer's should not be viewed as a single disease, but should perhaps be better referred to as a syndrome. And a lot of researchers have already written, especially in the humanities context, about Alzheimer being an extremely vaguely delineated medical category. And Laura Koik has also shown how this sort of has a very long medical history from the inception of Alzheimer's. Most problematically, perhaps, is that despite many decades of research, there is very limited knowledge of causes and neural mechanisms. Now, the currently dominant framework is the so-called a.r.t.e.s one, which posits that pathophysiological changes develop across a certain trajectory. So first they start with amyloid plaque accumulation. These are then followed by tau tangles, and finally this leads to neurodegeneration. And although it's a dominant, it's not a framework that doesn't have critics, so it's not universally accepted. And especially sort of the challenging aspect is that pathological roles of these plaques and tangles are not really known, and there is no straightforward correlation between any of these sort of pathological changes and the clinical manifestation, which means that it can happen a lot that healthy people die, and then on autopsy, it is sort of determined that they have a lot of these pathological changes, but they have never developed symptoms of Alzheimer's. And also in the other direction, some people apparently die on Alzheimer's, but the amount of pathological change that is found regarding plaque and tangles is not the one that should be expected. Now, the current diagnostic procedure in the clinical context is based on a battery of cognitive and neurological tests that assess cognitive impairment. So in that case, the patients already have sort of very clear-cut symptoms when they come to a physician to be diagnosed. And typically, these cognitive and neurological tests are supported by diverse neuroimaging and cerebrospinal fluid biomarkers of pathology. And sort of these additional biomarker tests are based on different types of neuroimaging, so positron emission tomography, so PET scans, and also different CFS tests. And they all aim to identify either these amyloid plaques or tau tangles that I have already mentioned. Additionally, non-invasive structural neuroimaging, primarily magnetic resonance imaging, so MRI, is used to identify anatomical brain damage. So it's this third aspect of the a.r.t.e.s model. And as you can see, basically all these sort of biomarker technologies are grounded in the a.r.t.e.s because they either sort of are aimed at A, T, or N aspect of the model. The problem is that all these tests basically don't have any standardized ways of combining them. There are also not clear cutoff points. So basically, the diagnosis is expert-based and is typically criticized in the medical literature as being too subjective, because in each case, the diagnostic physician has to sort of decide how many and which tests to combine and where to define the cutoff points. And the especially sort of troubling aspect is that in approximately 20% of cases, it is presumed that the diagnosis is inaccurate, especially in the early stages. This has basically two consequences. One is that many of the potential patients never get diagnosed, and some of them also get diagnosed with a significant delay when the brain damage is in already sort of late stages, and not only unreversible, but also untreatable. And even today, despite these sort of increasing number of biomarkers, you can find in the literature, medical literature references, that the definitive diagnosis is only possible after death. So when the histological measurements of plaques and tangles is performed. All these clinical differences and ambiguities and sort of problems have led to something which has been discussed quite a lot in the humanities-based literature, and Annette Leibing, who was also part of this series of talks, has written especially about it. So this is this preventative turn in the AD diagnosis, where there is an increasing shift to trying to diagnose patients who either have mild symptoms or no symptoms, and who are then defined as subjects of being at risk at developing AD in some future. And that basically means two things. One is a focus on increasingly developing new biomarkers, which would enable this sort of earlier diagnosis, where one is not reliant on already clear-cut symptoms. And the other aspect is the increasing reliance of a medical construct of mild cognitive impairment. And mild cognitive impairment is basically a kind of cognitive decline, which is not very clear yet. So the grounding assumption of this preventative turn is that MCI subjects, so patients with or subjects with mild cognitive impairment, could benefit from early therapies, which would be more sort of successful at preventing or slowing down progressive brain damage. Although MCI was basically introduced as a potential sort of solution for this delayed diagnosis, it could be instead argued that it's introduced sort of new problems, because MCI is even more difficult to diagnose, both through cognitive tests and through visual inspections of brain images, whether they are PET or MRI. So in a sense, as a potential solution, it turned out to be a highly ambiguous clinical category that is sort of placed in this gray zone between normal aging and clinically probable AD. And there's quite a lot of discussion in the medical literature about how to sort of categorize MCI. Is it already a sort of prodromal AD? Is it already part of the pathological picture? Or is it just a sort of a risk of developing future disease? And clinically, there's even bigger sort of ambiguity, because medical studies have shown that the annual conversion rate from MCI to AD is around 8 to 15% across different studies, so quite a sort of large variation. And while most subjects, so around 2/3, remain stable and don't develop AD even after 10 years, some even spontaneously revert to healthy conditions, so no symptoms at all. And according to some study, this sort of percentage of spontaneous reverters is quite high, around 20%, I found. And this is exactly where machine learning-based research on predictive diagnosis is sort of aimed to provide an answer. So it's being developed as part of the precision medicine's promise of developing targeted preventative treatments and answering the question that is currently sort of bothering the clinicians, which is how to objectively or quantitatively identify MCI subjects who will convert to AD. Now, initially, much of the sort of machine learning-based research, and I don't suppose I have to talk much about what machine learning is. We can do that in questions if that's sort of necessary. I can just say it's just a sort of a subset of AI. So the initial research was really sort of focused on differentiating AI patients from healthy individuals or patients with other types of dementia. And it is only recently, within the last 10 years, that a sort of a distinct new strand emerged within ML-based research into AD, which explicitly aims to develop ML-based algorithms for automated image analysis of brain scans to predict which patients will in future convert from MCI to AD. So this is sort of the driving question behind these studies. And basically this research has two distinct aims. One is some future application in clinical context for predictive computer-assisted diagnosis in still healthy subjects, so non-symptomatic, or MCI subjects, so subjects who have mild cognitive impairment, but are still sort of very ambiguous. And sort of the idea is that this research will produce new neuroimaging biomarkers, so kind of biomarkers that are based on neuroimaging data. And the second sort of slightly more distant, even more distant aim is that if such biomarkers could be identified, they could, in the second step, inform new research into AD's neurodegenerative mechanisms and support more targeted treatment development. So in a sense, it aims at both of these questions, right? So on the one hand, this clinical ambiguity of the early diagnosis in non-symptomatic or mildly symptomatic patients, but also at the fact that drug development and clinical trials of drugs fail at such a large rate. And the hope is that ML could somehow sort of turn that around. And this is really sort of important to emphasize that both of these aims in terms of applications are sort of in very distant imagined future, and there's no indication that at the moment any of the studies are developing something that could be soon applied in the clinics. Nevertheless, I argue that it's important to look at what is happening within this research at the moment. This brings me to the next step of the talk, where I want to sort of provide some basic insights into what this research is about and sort of how it's structured before I then go to the individual studies. But this framing is sort of important to understand how it functions. So basically, all of this research, all of these studies are both driven by and situated in the neuroimaging community, and they're all multidisciplinary studies. So typically, it's a combination of neuroimaging experts and computer scientists. Much of the studies so far have focused on high-resolution, three-dimensional MRI structural brain imaging. And the interesting thing is maybe also to mention, I said that MRI is also used in the clinical context when diagnosing. But typically, the kind of clinical scans that are used for diagnosis are very sort of low resolution and not necessarily covering the entire volume of the brain. So the kind of data that is being used for this research is typically sort of produced within the research context. So they are sort of really sort of high resolution and covering the brain without any gaps, which also sort of makes a difference to the clinical translation, because typically this is too expensive and too time consuming to generate such images to do it in typical clinical contexts. And the important aspect is that MRI in itself is a non-invasive technology, and it registers tissue loss that in AD is not localized, but really sort of widely distributed across the brain. There are some sort of anatomical regions that are particularly implicated in AD so far, but nevertheless, a lot of patients, like I said, have very sort of diverse topographic distribution of degeneration anatomical. So these images give insights into the distribution. Now, because it's ML-based, this research requires huge data sets to enable statistical modeling. And in a sense, the emergence of this research is also explicitly tied to the establishment of large-scale open access digital databases, the most important and the most widely used of which is the a.r.t.e.s, so the AD Neuroimaging Initiative dataset. But there are also some additional ones like a.r.t.e.s, like in Australia, and some of the studies also targetedly produce their own data. But sort of the first and so far the most influential studies have typically relied on a.r.t.e.s. What all these databases have in common is that they collect longitudinal multimodal data tracking progression of patients from MCI to early and even late AD. And the kind of data can differ from database to database, but typically includes at least MRI, PET, and clinical data. In some cases, it's also genetic data. Now, the guiding assumption that sort of drives this kind of ML research into predictive diagnosis of AD is the belief that there are latent visual patterns of specific structural brain pathology in MRI scans of patients with mild cognitive impairment who will in future develop AD. And these kind of patients are typically referred to in the literature as MCI converters. A sort of a second aspect of this assumption is that these pathological changes are absent in those MCI subjects who will not convert to AD at some point. And these differential visual patterns are then imagined to be predictive of future conversion to AD. Now, importantly, if we accept the existence of these patterns, we have to admit that they are entirely invisible to humans and that they can only be identified, extracted, and modeled by machine learning algorithms. Because the sort of assumption is that these pathological changes, if they are there, they're so minuscule that they cannot be seen by people looking at black and white images. I didn't sort of explicitly say which images are MRI images, but these are MRI images. And although experts can read a lot of details from the images and you can zoom in to them, and you can also sort of, because they're 3D, you can also drive through the brain of the patient to look at certain aspects. Nevertheless, some of the changes are simply not available to human visual inspection, even to a trained expert. And sort of the idea is that ML algorithms can statistically model such minuscule changes. Now, another sort of important aspect about this research in general is understanding its predictive scope or its predictive aims. So although it's called predictive diagnosis, what these algorithms are meant to do in future, it is really important to emphasize that this kind of predictive diagnosis is not about quantitatively estimating the likelihood of a given future outcome. So it's not about giving a percentage of chance that somebody develops or doesn't develop AD at some point in future. It's also not about sort of estimating at what future point this could happen. So this is not the kind of question that these algorithms are designed to answer. Instead, what these algorithms actually do is what is called as image classification problem. So basically what they do is that they are fed MRI images once they have been trained and tested. And based on these MRI images of unknown subjects, they are supposed to assign a particular predefined class or category or label to these images. So either sort of label the unknown data as a future IMC converter or as a future IMC non-converter. And also the important thing is that these algorithms are sort of designed to perform single subject prediction. So a lot of the neuroimaging research is typically done at the group level, where neuroimaging is also fed into different kind of algorithms, so not necessarily machine learning. And then in the research context, group differences between AD patients and healthy subjects are being identified. And these are then supposed to sort of provide insights into mechanisms of the disease. Here, this is not about group differences, but really sort of the ability of algorithms to assign a particular label to a single subject by classifying them either as healthy or as future patient based on MRI data. Now, the important aspect of these specific algorithms, and that's why they're also called machine learning, so is that these types of algorithms are not explicitly programmed to learn the classification, but they are trained on certain referential data sets. So as you see here, initial data set in the image, which are called ground truth. And these data sets are typically sort of split into training set and testing set, and algorithms are first trained on one set, and then once the training has been finished, then the models are tested on a testing set to see if they can accurately assign a diagnostic label to the data they haven't seen before during the training. So the idea is that during the training, they learn on their own a certain differential patterns between two classes, and I will talk later about what these two classes are. Let's say for the time being between healthy patients and healthy subjects and AD patients. And sort of then based on this pattern, which sort of should be differential, right? If they can then decide if MRI data from an unseen subject belongs to one or another category. Now, this search for the unknown predictive visual patterns, which then in turn, if validated, would be the wished for biomarkers, neuroimaging biomarkers, is exploratory in the sense of Friedrich Steilner, because it doesn't have any a priori theory about underlying neurodegenerative mechanisms. So it's just sort of a hypothesis that this difference should exist at the level of images, but what this is completely unknown. And it is also purely data-driven. Now, within the medical literature, also this neuroimaging medical literature on ML algorithms, the main focus is on improving what is referred to as accuracy metrics. So after the algorithms have been trained, then they are evaluated on the test data, and during this evaluation, their ability to correctly classify the unseen images is assessed through particular quantitative measures, and one of them is accuracy. There are quite a few different metrics, but accuracy is typically the one that is at the foreground of everything. And most of the studies typically aim at the moment at accuracy measures of 70, 80, some even 90%. And much of the discussion is basically within this literature about comparing new studies to previous studies and seeing if this accuracy metrics has risen or not. Sometimes even just a question of a few percentages. However, my claim is that such quantitative performance metrics gloss over unresolved past and present ambiguities in clinical AD diagnosis that gets implicitly inscribed into machine learning algorithms during this ground truthing process that I have just described. And this is basically the questions that I'm primarily focusing on when looking at this research. So I'm interested in how this future ML-based predictive diagnostic separability between subjects labeled as MCI converters and those as MCI non-converters is currently constructed at the level of MRI images through media-specific operations of transcriptivity. And this is what I will define shortly, what I mean by that. I'm also interested at the epistemic contingencies of this process of sort of defining this predictive diagnostic separability. And finally, I'm interested at broader consequences, both sort of epistemic and ethical, for future clinical conceptualization and diagnosis, as well as management of AD. So if these images are used to sort of redefine the difference between health and disease, what is the broader effect of that? And transcriptivity is a term introduced by the German media scholar and philosopher Ludwig Jäger. I will first read his definition of it, and then I will sort of rephrase it a bit. I find it important also sort of to refer to his initial definition, but it's sort of quite opaque in a sense. So for Jäger, procedural logic of meaning ascription through the processing of intra and intramedial differences as symbolic activities is transcriptivity, and it serves to organize the mutual reference of media to media. Semantic effects are owed to these interconnections. So in a sense, in short, what this means is that context-specific procedures by which meaning is constructed and communicated across different media shapes how data are used and interpreted. And this means that data as MRI images don't actually give us access to some existing reality out there. They cannot sort of tell us if there is a diseased brain or not a diseased brain in itself, independently from the mediation of the images, but sort of how we work with these images, how we produce them, how we relate them to other images, how we relate them to other concepts. So this intermedial and intramedial, intermedial is sort of relation between images and labels, and intramedial is relation between one type of image and another type of image. So how we sort of connect these images to one another and how we connect them to broader concepts and to maybe other kind of data and other kind of media, this is what gives us a production of meaning and then tells us if we think that there is a diseased brain out there or not. And we can never access the brain itself. And basically, my approach is drawing on this sort of processuality, and that's why I especially like Jaeger, because he insists really sort of that if we want to look at how meaning is produced in a specific medial context, then we really sort of have to unpack the processes and procedures through which this meaning was constructed step by step. And sort of my approach is to use then Jaeger and to analyze and compare method sections of various published research papers that have developed ML algorithms for predictive diagnosis of AD based on neuroimaging data, and to sort of analyze these procedures that they applied. And this means that my focus is not on inherent data biases, which are often sort of discussed in the humanities-based literature when they look at development of ML algorithms for medical purposes. It's also not on the opacity of AL algorithms, which is also another aspect which is sort of very intensely discussed, or on the performance metrics like accuracy, which is something that is discussed not so much in the humanities-based literature, but as I said, in the medical and computational literature itself. Instead, what I focus on are human decisions, especially in the sense of community of researchers. So I'm not so much interested in how a single person, single researcher makes a certain decision, but how sort of certain decisions within the community can be traced through the published papers, and how these decisions then shape the development of algorithms. Or simply sort of said, I'm interested in how researchers work with data. And although sort of this is my combination of STS and media studies, because a lot of STS investigations are then ethnography based, so they sort of follow researchers in concrete labs. In this case, I don't do that because I'm also, like I said, interested in the broader community perspectives. So following researchers within one lab would not provide me insights into the kind of questions that I'm interested in looking at. That's why I need a compilation of publications to be able sort of to look at certain developments and certain patterns at how the community judges the data to be useful or judges the methods necessary to deal with these data. And another important thing is also to say, when I talk about sort of decisions that researchers make during the development of algorithms, it's actually an entire chain in case of ML modeling. So there are quite a few different steps at which they have to make very far-reaching decisions. So one of them is the choice of algorithm type, right? So they use different underlying mathematical models. For instance, they can sort of use classifiers like, I don't know, like they can use classifiers or they can use deep models. So they're very sort of different ways of using different algorithms. They have to make choices about which training and testing data sets to use. Like I said, AD Neuroimaging Initiative is one that has been mostly used in a vast majority of studies. But in some cases, some of the researchers also generated their own data sets or did both, looked at ADNI data set and their own data sets. That's also an important decision that sort of affects what kind of algorithm you can produce. Another aspect is how many subjects, how big a sample size you want to have. And generally, most of the studies struggle with small data sets, but that's also sort of a normal development in most of the data-based sciences. Another aspect, because these are sort of imaging data, is how these images have to be pre-processed before they can be fed to the algorithms. And for instance, one of the pre-processing step is so-called normalization to a standard space, which means that because we all have different brains, they first have to be warped and made comparable. And how this is done algorithmically also influences what in the end sort of comes out. Another decision is which visual features to compare. So it's not just that these images are sort of compared directly one to another via the algorithm, but typically they are submitted to rather sort of complex analysis where certain regions are selected, or cortical thickness is first computed, or certain volumes of certain brain regions, or shapes or textures. Textures are first sort of computationally defined, and then these features are fed into algorithms, and then the algorithms sort of compare them. Another important aspect is how to define classification tasks during ground truthing, which I will come to shortly. And an equally important decision is sort of validation choices. So which test data to use and which performance metrics. Now, what I particularly focus on, and that's why I want to sort of to present this entire chain, is actually the classification task during ground truthing. So relatively minor aspect of the entire chain. But still, I argue that especially this aspect has crucial sort of semantic value and presents also a crucial semantic problem, because the attribution of medical meaning is what happens at this stage. So what basically the community, the scientific community, is currently struggling with is how to organize the comparison between different classes of MRI images to enable sufficient predictive diagnostic separability between converters and non-converters, and identify these assumed differential visual patterns. So, as I said, at this stage is where this transcription of assumed, I'm sorry, of assumed differences between future disease and health is taking place at the level of MRI images. Now, based on a close reading of method sections of around 30 studies that were published between 2010 and 2024 on ML algorithms for predictive AD diagnosing using explicitly MRI, I basically identified 5 widespread approaches that researchers use to define classificatory comparisons. And I will now present all 5 of them. So the first comparison, which I call AD versus healthy controls, is the following. Basically, algorithms are trained to classify brain scans stemming from AD patients versus healthy controls without using any data from MCI subjects. In the next step, the algorithms are then tested on MRI data from MCI subjects to predict if these will convert to AD. So they're not trained on MCI subjects, but they are meant to predict conversion. And the guiding assumption, although very sort of implicit and not explicitly stated of this comparison, is that binary comparison between healthy controls and AD patients is largely equivalent to the comparison between MCI converters and MCI non-converters. And this is basically the comparison that started much of the research and was especially sort of dominant in the early studies, but I found even some quite recent studies that continue to operate under this rather simplified assumption. The second comparison I refer to as MCI only has the following structure. So both the training and the evaluation of ML algorithms is based on using baseline MRI scans from MCI subjects who are retrospectively categorized as converters or non-converters based on later AD diagnosis. Now, this sounds maybe a bit complicated, but the point is that none of these open access databases have categories or labels such as converters or non-converters. They simply scan people who are at the beginning MCI subjects, and they either sort of subsequently remain MCI subjects or they convert to AD. And basically, these researchers, these study authors, when they go to these databases, they then explicitly search for those subjects who converted and who didn't convert, and then go also and use baseline scans. So baseline scan is the first scan made of this person when they became participants in the studies that result in the databases. And the distinction between AD patients and healthy control is entirely neglected when training these algorithms. But the semantically critical decision is defining this cutoff period. So when do they decide that an MCA subject has become a converter? And this differs extremely between studies. Some define 18 months, some 24 months, some 3 years. But some recent studies indicate that maybe the optimal cutoff point should be 5 years. There is actually no consensus, and these decisions are highly ambiguous because it means that studies who defined a cutoff point of 18 months may be actually defining people as non-converters who later, at a longer cutoff point, became non-converters. So the third comparison is what I call longitudinal trajectories. These kind of comparisons use repeatedly collected data from the same individual over time. And basically, the classification compares the trajectories that trace this conversion from MCI to AD. Here, the underlying assumption is that conversion from health to disease is gradual, and that the only way to reliably predict conversion is by modeling this gradual trajectory. However, again, there are also inconsistencies here how these trajectories are defined. First of all, studies use very different number of serial scans. Some use, for instance, only 3, others use much more per subject. Also, there's a big difference in intervals between scans. Some define intervals as 6 months, others as 12, some also as 24 months. And the conversion period is basically similar to this cutoff period in the previous comparison. So when do they sort of stop looking at the trajectory? Again, here variations are between 18 months and 5 years. So in a sense, by making these decisions, they automatically have very different concepts of what counts as a comparable trajectory of a disease. The fourth comparison is what I refer to as multi-comparison approaches. And in a sense, this kind of comparison has different sub-comparisons within it. The idea here is not to use single binary distinctions like in the first two comparisons, where either patients and healthy subjects were compared, or converters or non-converters, but to use multiple comparisons. And one example is so-called multitask learning, where algorithms are trained simultaneously on different comparisons. So MCI non-converters versus converters and AD patients versus healthy control. So algorithm looks at patterns that are important simultaneously for these comparisons. Another example is so-called transfer learning, where algorithms are first pre-trained on one comparison, typically AD versus healthy controls, and then fine-tuned to sort of do the comparisons between MCI converters and non-converters. But in this case, this is different from the first comparison, where just sort of evaluating was done on a different comparison. Here's really sort of additional fine-tuning training done on this second comparison. And here the underlying assumption is that the differences between non-converters and converters may be too subtle for any sort of direct binary comparison, and that similar or related comparisons are necessary to provide additional information. And this brings me to the final fifth comparison, which also unites different multimodal approaches. So in this case, although MRI data remain central, they are combined with either with other neuroimaging or non-imaging modalities. And majority of studies so far have done binary combinations. So in addition to MRI, for instance, PET or fMRI, so another sort of functional neuroimaging. What more recent studies have also done is that they really sort of do an entire multimodal integration where MRI is also sort of combined with CFS levels, with genetic data, with cognitive scores, with demographic information. So sort of clinical aspects are really sort of combined with these various potential biomarkers. And here the assumption is that MRI data alone are not sufficient for predictive diagnostic differentiation. So in the literature, if you look sort of at the studies themselves, these choices which comparison to make are often driven by purely pragmatic motivations. So a lot of times they depend on the type of algorithm that they are using and sort of which kind of comparisons work the best. Some study even sort of employed different comparison within the same study, right? And then they test which comparison produces best performance metrics and then use that comparison as a sort of best for modeling the algorithm. So in a sense, the decision on how to delineate disease and health is largely guided by statistical efficacy in image classification and technical consideration rather than medical interpretability. However, what I have sort of tried to also show by analyzing these different comparisons is that they establish very different semantic relations between health and disease. Some imply very clear, delineated binary disease and health states. So first and second comparison, right? MCI converters are the same as AD patients, and MCI non-converters are basically regarded as healthy. Others, like the third comparison where we had this longitudinal trajectories, imply a continuous disease spectrum and already have this idea that any MCI is likely to have some pathology. And if you wait long enough, this subject will also become a patient. Pure MRI studies basically equate illness with neuromorphological changes. So ID is primarily brain-based and anatomy-based, whereas the subsequent multimodal studies basically presume multifactorial disease mechanisms and argue that all these interactions are important for diagnosis. And there are additional sources of contingency, because the basic assumption is that MRI images are unambiguously labeled. So everything that goes into ground truthing, both for training and testing, is assumed to be unambiguously labeled. But as I showed at the sort of introduction, these labels are derived from clinical context and uncertain diagnosis, mostly not verified post-mortems. And as I also sort of noticed when I was talking about the second comparison, MCI converters and non-converters are retrospective labels that are assigned to earlier MRI scans by these studies. And any MCI non-converter could later convert, depending on how these cutoff points and trajectories were defined, which makes the distinction also temporally unstable. Now, the sort of the problem is that all these implicit assumptions and epistemic contingencies get inscribed into algorithms during ground truthing. And that means that even if algorithms appear to have exactly the same accuracy and specificity metrics, they might, in fact, define very different boundaries between disease and health, depending on which comparisons underlie their production. So in a sense, what I argue is that there's a large discord between these seemingly objective quantitative accuracy metrics that the study is always foreground and the semantic ambiguity. So what are these biomarkers of? Which just brings me very briefly to broader ethical implication that I just wanted to touch upon. So what these studies do, in my opinion, is that they very much reinterpret MCIs, subjects from sort of vaguely defined intermediary state into one where algorithmically detectable disease-specific neurodegenerative changes are already assumed to have occurred, because this is sort of the idea about this sort of present patterns that ML algorithms are supposed to detect. They also implicitly homogenize MSI because they only look at the progression from health to AD as nearly linear and one way. All MCI subjects in the databases who have these wayward trajectories or who recover spontaneously, and I mentioned at the beginning that some studies say it's up to 20% of MCI subjects, they are not included in ML-based studies. They're really sort of explicitly excluded as outliers, so they're not used for training. Another implication is that neuroimaging data in most of these studies is entirely divorced from diagnosis, which is basically also the idea, because they want to sort of be used in patients who are non-symptomatic. But nevertheless, this divorcing from symptoms aligns these studies with this broader shifts towards a purely biological definition of AD, where symptoms become less and less in the focus of medicine. And also the quantitative performance metrics really overlook all these epistemic contingencies that I have tried to delineate and that are especially important during ground truthing. Which brings me to the conclusion, sort of just briefly to summarize what I've touched upon. AI image classification algorithms for predictive diagnosis of AI using neuroimaging data is explicitly aimed at displacing what in the medical literature is always criticized as subjective and expert-based clinical diagnosis, implying thereby that these diagnoses are unreliable. However, I have tried to show that this emerging research is nevertheless shaped and constrained by current ambiguities of AD and MCA in medical practice, because the data that they use are produced by these clinical practices. And I have focused on how human creators inscribe epistemic contingencies into algorithms during ground truthing through ambiguous definitions of boundaries between disease and health, by sort of not having a clear medical definition of where the difference is, but basically relying on statistical solutions and technical solutions. Future-oriented visions of algorithmic separability, in sense, already perform a subtle reconceptualization with AI of AD with multiple ethical consequences that I touched upon briefly. And basically what I want to suggest is that these issues really sort of need to be addressed at the conceptual level. So it would be really sort of necessary also for researchers to think when they do these comparisons, what are they actually medically comparing and how are they redrawing the boundaries, and not just sort of these technical and computational solutions that look at increasing accuracy of classification, but classification of what? Thank you.